My name is Barry Connors. I'm the chair of Neuroscience here at Brown. And my main job will be to introduce the introducers. And I'm going to start by introducing Ed Harrott, who is the Alva O. Way University Professor of Medical Science, Associate Dean of Medicine and Biological Sciences here at Brown. And he's going to give us some opening remarks before we kick off with the main show.
Thank you, Barry, and thank you all for coming to this exciting day of lectures and discussion, and culminating the week of stellar presentations and again discussion and the exciting format of the Sweatbox, where students can dive right in and query the luminaries in the field about the underpinnings of their pronouncements and scientific conclusions. So I stand here today to represent Jack Elias, who is our Dean of Medicine and Biological Sciences, who unfortunately couldn't be here today because of other scheduling conflicts, and I represent the Division of Biology and Medicine, which, as many of you know, is the faculty of which is engaged in the education of not only the medical students, but also the undergraduate biology concentrators and the neuroscience concentrators. And those undergraduates together account for about 20% of the graduating class every year. And so we're excited to have and I'll say a few more comments about some of the contributions that the undergraduates have made in areas that we're very interested in. In partnership with the faculty in computer science many years ago, over 25 years ago, the biology faculty and computer science faculty organized an undergraduate concentration in computational biology, which is one of the earliest, if not the first, such major undergraduate concentration in the country, and that's still going very strong. And it's also been expanded into the graduate area so that we have within the Division of Biology and Medicine, we devote some of our resources to support graduate slots in computational molecular biology. And that particular program is organized under Will Fairbrother, who is a member of the CCMB, Center for Computational Molecular Biology, and Department of Molecular Cell Biology. And I believe they filled their slots this year. And overall, in fact, we've had a very successful year of graduate recruiting in the Division of Biology and Medicine. We, at the latest count, have 44 students who will plan to matriculate in September and join our various programs, and there may even be another one we're going to accept due to a computer error on CollegeNet. He emailed us and he said, "I hit the yes button," and so we're dealing with that right now. But that group of 44, so we're bringing in 44 students overall in our 8 graduate programs, and those account for, that's actually setting a record for our Division of Biology and Medicine. That's the largest class we've ever brought in. On a related front, I'm happy to announce that we have recruited a pair of new faculty into the medical school who will lead the effort on biomedical informatics. So again, we in biology really understand the importance of computational approaches, and now with the latest advances, the legislation and the extension of Obamacare across the country, and there's now a larger, greater incentive to utilize the massive amount of data that is being accumulated in the healthcare system. And our partner institutions, Lifespan and Care New England, are going to be introducing electronic medical records. Lifespan has already done that as of April. They went big bang, and all their clinical facilities are now using Epic, the system called Epic, and which technologically, I'm told, is not terribly advanced, but it's now become the standard across most of the country, and about half of the academic medical centers in the country are using this system. So there's going to be a huge store of electronic medical records that, in theory, should be accessible and could be the subject for not only improvements in healthcare delivery, but also leading to new hypotheses about disease causation and highlighting potential scientific questions that our basic science faculty can address as well. So we're excited about these two hires. They're starting in July 1. They both received their PhD. It's a husband and wife team, and they received their PhDs a number of years ago at Columbia in their PhD program in biomedical informatics, and they've spent the last few years running the Center of Biomedical Informatics at the University of Vermont Medical School. So we're looking forward to that. They're also going to be very much engaged in the Rhode Island Quality Institute, which is a statewide effort, and it's a health information exchange. And I don't know how many of you know that about half, just close to half the people in Rhode Island have signed up to share their electronic medical records for research purposes and for emergency care, so that these 440,000 people, if any of them require emergency care, the attending physician in the state of Rhode Island, at any facility, can go online and look up their medical records, look up their their lab tests, their MRIs, their pharmaceutical, their medication history through CVS. So CVS is a partner with us, and so this is the only health information exchange in the country where it's the entire state is involved in this. And recently, I heard that Connecticut had a similar effort that was funded a number of years ago through the Health Information Technology Program, and they've basically given up. And some of the counties in Northwest Connecticut are now asking our Rhode Island Quality Institute to step in and take over and run their effort. So one of our I know I'm going on a little bit on this the sort of the big data in healthcare, but I think this is very much in line with the underlying theme of this week in terms of the importance of computation, and I think these electronic medical records that are going to be available to probably more with some variability across the country, but I think Rhode Island has some unique characteristics to it that will allow us to take much more advantage of it. And I think we have trained a lot of the expertise in this area as well. There's a new department of biomedical informatics at Harvard Medical School that's going to be starting July 1, when it's going to have 5 faculty. The inaugural chair of that department is someone named Zach Kohani. Zach Kohani was an undergraduate here at Brown and did a double concentration in biology and in computer science. And then he went on to do his PhD elsewhere, got his MD and PhD elsewhere, and then is now heading up the Harvard Medical School effort in biomedical informatics. A week from tomorrow, on the commencement weekend, I think the so-called Ruth Sauber lecture will be given at around 9 o'clock in the morning next Saturday, a week from Saturday, as part of the commencement weekend. And the speaker will be Atul Butte. Atul Butte is an MD, PhD. He was a computer science major here at Brown, and then he was part of the PLME program, which means that he was part of that 8-year program, which led to an MD from the medical school here. And most recently, he was leading the biomedical informatics effort at Stanford up until about 3 months ago when UCSF recruited him away from Stanford to head up the entire University of California biomedical informatics system. So he's going to be speaking here a week from Saturday as part of his reunion class, medical class. So Brown has certainly been noteworthy in terms of making major contributions in this area in terms of training the undergraduates, and we really look forward to the next stage and bringing in faculty expertise now in biomedical informatics. And we have a large grant application that we've submitted through the division, which was very multidisciplinary and integrative. It's a proposal to set up a Center for Clinical and Translational Research, and it's a $20 million award if we're so fortunate as to receive it over 5 years. And one of the key elements of it is to enhance the composition and strength of a Center for Biomedical Informatics, and to build a really strong bridge with the Rhode Island Quality Institute, so that we would actually have an employee within the firewall of the Rhode Island Quality Institute who would have access to all that data. So I think that's about everything I wanted to say in terms of giving some plugs for what Division of Biology and Medicine has been contributing over the years, and we're very supportive of efforts in enhancing the various applications of computational approaches to scientific advances, and we really appreciate Soren and his team for organizing this symposium. I've been to each of the two previous ones, and they've been exciting, and this one has also continued in that tradition, and I really look forward to the next one now that we're about to finish this one. But I look forward to the exciting speakers this morning, and I'll pass it back over to Barry.
Thanks, Ed. Okay, on to the brain and neuroscience. And I've asked Jim McElwain to introduce our first speaker. Jim is a colleague of mine, Professor Emeritus of Neuroscience, former Fox Chair in Ophthalmology and Vision Sciences, and a close colleague of David Berson.
Thank you, Barry. It is tempting to think about the eye as developing from a single Ur cell that flowers into the organ of sight that we know, but that's not how it happens. The eye is actually constructed, assembled during an elaborate developmental process that brings together tissues from different genealogies, and one of these tissues is the neural tube, or piece of the brain, the developing brain, that is, as it were, exiled, banished to the periphery to live inside this organ that sits in the orbit. This bit of brain comes out there, and it forms eventually several things, including the retina, which is the topic of this morning's talk. The retina, this piece of the brain that's been exiled to the periphery, stays in touch with the brain that's stuck inside the skull by virtue of some pathways that are occupied by what are called retinal ganglion cells. You're going to hear much more about them as we go along. But the retinal ganglion cells have been the subject that has occupied Dr. David Berson, who is our speaker, and his team. They have made fundamental contributions to our understanding of what these cells do, how many types there are, what their physiology is, their connections, and so forth. And some of these contributions are, in fact, groundbreaking, even revolutionary. The team and Dr. Berson have been recognized widely for this, and I just will mention two recent examples of this. Last year, David received the Brian Boycott Award, named for one of the premier retinal neuroanatomists of the 20th century. This is given by the FASEB, and for work that has advanced our knowledge of the retina and its role in vision. Just this year, he received the Friedenwald Award from the Association for Research in Vision and Ophthalmology for outstanding contributions, outstanding research that contributes to basic or clinical knowledge related to ophthalmology. So given that, I'm sure you want to hear what Dr. Berson has to say about the brain that is in your eye. So it gives me great pleasure to introduce my colleague, David Berson.
We got the lavalier. What Jim didn't say was that I was a postdoc in his lab for 3 years, and I would not be standing here today if it weren't for the training that he gave me. So I'm deeply grateful to him for not just the introduction, but for all of the instruction and inspiration that he's given me back then and through the years since. He still comes to my lab meetings, which is a great boon for my students, as I'm sure they'll tell you. Okay, so as Jim has said, the retina is actually a piece of the brain, and here's this developmental process that he was describing. Here's the early embryo, and on this lateral side here, you can see the beginning of the optic cup forming. And if we were to take a slice through this embryonic neural tube, this developing nervous system, you can see that, in fact, the retina, which is right here, is in direct continuity with the brain. In fact, it is a piece of the brain that has been pushed out into the periphery. And this, in a sense, is our window on the world, but in fact, the world can look back in through the front of the eye and see what's going on, at least in this one piece of the central nervous system, this piece of the brain. So even with simple ophthalmoscope images of the retina through the front of the eye, you can actually see the blood vessel pattern and so forth, but you're actually looking at the surface of a piece of brain. There are many imaging methods that have been devised to allow you to see much more detail than is possible with a simple ophthalmoscope. And one of these is a method known as OCT. It's actually a method that allows you to see the layers of the retina. I'll have much more to say about the layers as we go along. But in addition, there are methods now for visualizing individual cells within the living human retina. This is known as adaptive optics imaging. And what you can actually see is the cone mosaic, the 3 different kinds of cones arrayed out on the surface of the retina, and their distribution cone type by cone type. So this is a very convenient piece of the brain to be working with, as it's visible even in an intact organism. Now, the real payoff, of course, is if you begin to remove the retina from the eye and put it in a dish. Now you're in a position to actually manipulate the cells there in multiple ways that allow you to piece together the circuitry of the retina and the processing that's going on through its various cellular layers. So the challenge here is to think about what's in store in the next 250 years in my case, in vision science. And that's a tough order, but I think there's maybe a way to go about this, which is to roll the clock back 250 years and see where we were back then, see all the ways in which things have changed in the intervening years, and then maybe try to do an extrapolation out to the future to see where we might end up. Fortunately, already 250 years ago, 1764, we knew that this theory, the emission theory of vision, was wrong, that something came out of the eye, some form of energy came out and played across the surface of substances in the world and told us somehow magically what was out there, something like the X-ray vision of Superman. So this was an idea promulgated by Euclid and a lot of the other classical natural philosophers. And it really wasn't until the advent of Arabic and Islamic science that it became clear that that was really not the way that it worked at all. And in fact, it was photons entering the eye. Of course, I didn't know about photons, but light entering the eye and somehow interacting with our bodies that underlay vision. Now, in this case, you're looking at Al-Hasan, who is one of the Arabic natural philosophers who figured out that this was, in fact, what was going on. And really, Al-Hasan's insight came from his own interest in optics, because he was studying the stars. He was looking up at the sky, understanding how optics worked. He recognized that the eye itself was an optical instrument, and that this must somehow create an image of the visual world inside our eye. Now, Al-Hasan knew that the image should be upside down if it was a standard optical instrument, and that bothered him. So he actually kind of put his finger on the scale and flipped it back over again, not quite knowing how it worked. This didn't seem to bother Kepler, who was another early astronomer who understood a lot about optics and was interested in the way the eye worked. And basically what Kepler said was, yeah, it's upside down on the retina, but somehow we figure it out anyway. It's not really a problem. And I think in that he's absolutely right. So here's just an image of this inverted representation of the external world on the retina. Of course, the brain figures this out. So we knew about retinal images, and Descartes was really the one who pushed the idea that it was actually the tissue at the back of the eye that was the key sensor here. And Descartes' view was that this sensory representation was somehow carried forward through the fibers of the optic nerve, the connection between the eye and the brain, to allow the soul to somehow appreciate the nature of the external world. So in Descartes' idea, the projections led from the retina back into the brain, and he was a little fuzzy on exactly where these terminations were. He actually was thinking that they might be the ventricles of the brain. But the key insight that he made was that the projection, the carrying forward into the brain of the retinal image was reinstantiated in a new representation. And so you can see this retinotopic or topographic mapping from eye back to the brain in Descartes' view. And this is obviously a very key insight. So the earliest idea of representations in the brain of the visual world dates back as far as Descartes. And this is pretty much all we knew as of 250 years ago. So everything that we've learned since is obviously huge. And if the process that we're looking at now, the advancement of techniques and approaches, different fields bringing their expertise to bear on this problem, continues to accelerate as we think it has. Up to this point, it's going to be really hard to imagine where we'll be 250 years from now. So just by way of sort of introducing the work that I'll tell you about what's going on in my lab, let me use some of the key things that we've inferred in the last 250 years as a way of bringing you up to speed on the key parts of the story here. So the first is that the retina is not simply a sensor. It's actually got some internal structure. And this was obvious already to Treviranus in 1830 or so. And you can see with his earliest histological studies of the retina, in this case from the crow, that the retina is a highly laminar structure. There are different elements at different levels, and this is key to understanding its organization. Now, for Treviranus, the idea was that light would come in and strike the inner surface of the retina, and that would be the only logical place to put the sensory elements that would detect light itself, and that somehow these sensory elements would then carry fibers back into the optic nerve, into the brain. Now, he got a lot of things wrong here. The optic fibers actually run over the surface of the retina, and as you'll hear in a moment, the sensory receptors that respond to light are actually deep in the retina, as far from the incoming light as they could be. Although, as you'll hear later in the talk, Treviranus was right for the wrong reasons in at least one respect. That is, that there are some photoreceptors in the inner retina. The recognition that photoreception, the absorption of photons and the conversion into a neural signal actually occurred in the outer retina, as far from the incoming light as possible. It was made by Heinrich Müller in about 1850 or so. And what Müller saw when he took fresh slices of frog retina was that there was some color out in the outer retina that he surmised might be the pigment that absorbs the photons and leads to this photosensory process. He then followed this up with an extremely clever experiment in which he visualized the shadows of his own retinal blood vessels as he moved a bright light around in the periphery of his visual field. And he worked out through simple trigonometry the apparent angular extent of the motion in the visual world for a given angular movement of the illuminating source of light, and worked out that the depth of the photoreceptors had to be far enough away from the blood vessels that it had to be in the outer retina, where we now know the rods and cones lie. Now, a major breakthrough in understanding retinal organization came with the advent of methods for staining individual retinal neurons. This is the Golgi method. There were several other methods as well, but this was the one that really broke the game wide open. And the scientist who really deserves the greatest credit for exploiting this method to allow us to understand the fundamental organization retinal circuits is Santiago Ramon y Cajal, a Spanish neuroscientist who eventually won the Nobel Prize along with Golgi, who was the man who devised the method that Cajal used. And one of the key understandings, of course, of Cajal was that the retina was made up of individual neural elements connected together by synapses. That is, that we're not dealing with a syncytium of cells, a connected network, but rather with individual elements, units that are communicating through what we now know to be both chemical and electrical synapses. So from Cajal's work, we began to understand the basic canonical retinal circuit from photoreceptors, which you see at the top, the rods and cones, as we call them now, through an intermediate neuron, the bipolar cell, and then eventually to the ganglion cell, sitting at the innermost layer of the retina. And these are the output cells. So these are the ones that give rise to those axons that run across the surface of the retina and then eventually punch back through the eye itself to form the optic nerve running back to the brain. So this is the canonical circuit. There are several other interneurons here, but the thing to recognize is that there are two layers where these synapses occur, and we'll be focusing in some detail at the inner of these two, the ones between bipolar cells and ganglion cells, and the modulation that occurs at that level. Okay, so now I need to try to introduce for you a fundamental physiological concept, which is the notion of the receptive field. So if you imagine one of these bipolar cells, which is carrying the signals from the photoreceptors to the ganglion cells, you have to understand that the processes from which it receives its input from these first-order photoreceptors extend widely enough in the retina that more than one photoreceptor talks to that bipolar cell. There's a region of the retina which, when activated by light, will make this bipolar cell respond. We call that the receptive field. And this can be defined in retinal space here. How far across the retina can you be and still activate your bipolar cell? But one could also as easily define it in visual field coordinates because of this isomorphism between the visual world and its projection onto the flat retina. So any given neuron in the retina, or for that matter elsewhere in the visual system of the brain, will have a receptive field, an excitable region. Now, the receptive fields of bipolar cells and higher order cells in the retina are actually not quite as simple as shown here. And this is due to the interaction with inhibitory interneurons at several levels of the retina, one of which is shown here, the horizontal cell. So this is an inhibitory neuron that spreads laterally in the retina and can cause a bipolar cell driven by excitatory input from its own receptive field center to actually suppress that response when there is stimulation out here beside the central region. So we now have an antagonistic center-surround organization, which actually is a very clever design for a contrast-enhancing transformation. So just to give you a quick sense of that, this is the work of Keffer Hartline and Steve Kufler back in the 30s through the 50s. This kind of lateral inhibition, center-surround organization, ensures that these output neurons are actually conveying not veridical information about the light intensity, but in fact are doing kind of a spatial derivative of that. They're looking for changes in illumination in different regions of the visual scene. So if you scan a light-dark contrast border like this with center-surround antagonistic units, the output of the cell actually ends up giving you sort of an enhanced version of this so that you can really, if you imagine the brain looking at the array of these cells, the brain would be much better able to detect the contrast boundary than it would were the cells without their receptive field antagonistic surrounds. So you can see yourself the perceptual consequences of this. If you look at an image like this, and at least under the right lighting conditions, you may see a kind of scalloping in the luminance distribution there. In fact, that's entirely happening in your own visual system. If I just isolate this third bar from the left by itself, you can see that when it's not up against the other bars, that you don't see that scalloping. So essentially what's happening is these contrast-enhancing elements in your visual system are lying to you, in a sense, about the number of photons coming from that region of the visual field in order to allow you to see that boundary between the stripes more clearly. It also accounts for the funny little dim spots that you may see at the intersections between the white grid in this figure as well. And I won't spell that out for you, but it's basically the same phenomenon. Okay, so the emphasis here is on the output of the retina, what the retina is telling the brain. And when we think about that, we need to be talking about the ganglion cells. As I said before, these are the cells whose axons make up the optic nerve. And already at the time of Cajal in the late 1800s, it was clear that these ganglion cells were incredibly diverse. So if you look carefully, you can see that there are many cells stratifying near the top of this synaptic layer in which they get their input from the bipolar cells, some towards the bottom, some spread widely across the retina, some more narrowly. And Cajal was aware that these were fundamentally distinct types of neurons, although they shared the property of communicating with the brain, although he had very little to say about what might be different in terms of the function of these cells. Here's another view of this. This is now taken from the macaque retina from the work of Dennis Dacey. And again, you can see the cells schematically shown in side view here as Cajal demonstrated it. And you can see the wide range of different stratification levels. Some are stratifying in more than one level, some only in one. And you can also see, maybe at least those of you close enough to the screen, that the branching pattern of the cells looked at en face also varies widely. Some are sparsely branching, some much more densely so. Now, the reason the stratification matters so much is that the lower order retinal neurons that interact with these ganglion cells themselves make synaptic contacts at different levels within this plexiform layer, this synaptic layer. So here are the bipolar cells at the top, and you can see that different bipolar cells drop their axons down to different levels. This suggests that there are different channels conveying information from the rods and cones to different subsets of ganglion cells by different populations of bipolar cells. Diversity is even greater when you look at the other class of inhibitory neurons, the amacrine cells. These are neurons of the inner retina, like the ganglion cells, but they come in a huge variety of different forms. The guess now is somewhere on the order of 40 in a typical mammalian retina. So obviously, these synaptic interactions are complex, and any individual ganglion cell has the potential to look at a modified version of the low-order retinal image as viewed by the photoreceptors through these complex synaptic networks. So we anticipate that there'll be a lot more functional diversity than simply the kind of center-surround organization that you've seen so far. The other thing that I wanted to mention about the anatomy of the ganglion cells is that they tend to form these very regular mosaic-like arrangements, so that the locations of the cell bodies tend to be non-randomly distributed. They tend to be separated from one another by a characteristic distance, and then the dendritic arbors spreading across the surface of the retina tend to have a arrangement in which they're essentially covering the entire retina efficiently, and in many cases are essentially tiling, efficiently covering that surface. And what this means then is that any given ganglion cell that is viewing the world in a particular way, and I'll have something to say about what I mean by that in a minute, every one of these classes of ganglion cells is seeing every place in the visual field. So we have multiple representations of the visual world embodied in the array of ganglion cells of different types. So what about the function? So one of the key distinctions between ganglion cells has to do with receptive field size. That is, how much of the visual world does any one ganglion cell see? In other words, what is its window on the world, right? It doesn't see the whole visual world. It sees just a small region related to its dendritic arbor and the photoreceptors that excite it. So we have large ones, we have small ones. Another fundamental distinction is that some ganglion cells are excited by increments in illumination, others are inhibited, and still others are excited by either increments or decrements. So these are the on-off and on-off type ganglion cells. We'll have more to say about what builds that distinction a bit later. There are differences in temporal coding as well. Some cells respond only when there's a transient change in illumination. So for a step in light, the cell will briefly spike and then shut up. Other cells carry a consistent representation of the level of light intensity, tonic or phasic cells. And then there are a bunch of very surprising highly specific features that you see among different ganglion cells. Some, for example, might be selective for the direction of motion of a contrast boundary across the receptive field. Others might have a very high rate of resting discharge and only be suppressed by activation of the receptive field. Some are orientation tuned. There are cells that are wavelength sensitive, that is their color opponent, and so on. So we have a huge variety of functional types that presumably map onto these morphological distinctions. And then when we trace the projections of these ganglion cells back to the brain through the optic nerve, we now recognize that there are dozens of distinct targets in the brain. These clearly have different functional roles to play. In some cases, these are very well established. In many of these smaller nuclei, not so much as known. But it's clear that some are involved in oculomotor functions, still others in high-level perceptual functions, in regulation of sleep and circadian rhythms, and so on. So this is more or less where we stand now. We understand that there are distinct ganglion cell types, but we don't really fully understand how many there are. We know that ultimately, we will be able to relate particular ganglion cell types to particular target structures and to understand their physiology in terms of what they're doing for the visual system. But our ability is, at the moment, is somewhat limited, and we haven't really gotten as far down this road as we would like to. We don't understand really all of the synaptic circuits that underlie the specific response properties or receptive field properties that individual ganglion cells have. We're beginning to reconstruct these circuits. In some cases, pieces of the story are understood, but much still to be done. And ultimately, of course, you'd like to understand the circuit in totality. You'd like to be able to understand each transformation that occurs at every synaptic level, and that clearly is beyond us at this point for most ganglion cell types. So this is really the focus of my own research program. So if we're going to go about this, if we're going to try to relate ganglion cell types, their physiology and morphology and projections to functional roles, how do we go about this? And I think for the most part, the way the field has approached this is to describe as many properties for as many ganglion cells as possible and to try to think about what those properties might be good for. That's a perfectly appropriate sort of bottom-up brute force approach that I think can be very valuable, and I've engaged in it in a major way in my own research program. But I think there's an advantage to reversing the question and kind of flipping it on its head and saying, let's just start from the point of view of what aspects of vision are going to allow us to survive in the world and reproduce and carry on our genes to the next generation. And that is taking essentially a Darwinian approach to this. What is it that animals use to survive in the world, find their mates, and reproduce successfully? What is sort of evolutionary fitness in the context of the organization of the visual system? And in the context of sort of vision and evolution, it's useful to revisit something that Darwin himself said about the eye. So he was blown away by the gorgeous complexity and articulation of this biological optical instrument. And he confessed, as you see in this quote here, that although he was obviously a big believer in his own theory, that if there were anything that would give him the willies and make him worry that maybe he had it wrong, it would be that the eye was so beautiful that it almost had to be designed. Okay, so I think this is very much an anatomical viewpoint, and it's very much thinking about the eye as an optical instrument. But I think it can be extended to the retina as an evolved biological structure designed to help animals survive in the world. And I think it can be extended into the brain. I think it can also be carried over to a thinking about physiology, about circuits and circuit functions. Now, one obviously would ultimately like to understand the physiological basis of visual perception, and many people are working on this. But this turns out to be a tough problem, and I owe Jim McElwain a lot for pointing out to me the tremendous utility of working instead in an aspect of the visual system that is more cut and dried, hardwired, and you might say reflexive, where the relationship between sensory input and motor output is more straightforward and where one might have more tools to be able to try to unpack this whole thing, to relate this to specific neurobiological phenomena. And of course, when we're dealing with reflexes, the great granddaddy we all have to sort of bow down to in this role is Sir Charles Sherrington, who worked on the spinal cord back in the early part of the 20th century, working on spinal reflexes. Sherrington himself had really very little to say about the visual system. But what he said about spinal reflexes, some of which is listed here, I think really does relate very well to this attempt to try to bring reflexology to a study of the visual system and its sort of integrated organization. So he said that reflexes, I'm paraphrasing here largely, reflexes are dedicated circuits. So you need to start with a sensory input. Ultimately, you're going to have a motor output, but somewhere in between, you've got some set of interneurons. There's some modulation. There's some integration going on there. And that when you're thinking about a reflex circuit, you want to think about the whole thing, start to finish. He said that reflexes are purposive in a Darwinian sense. You know, this is kind of interesting because Sherrington comes at a time only 50 years after Darwin's theory is promulgated. And after a long period where many physiologists were sort of waving their hands about the purposes of behaviors and reflexes and so forth, and thinking about these in teleological terms that made a lot of people very nervous. So this is, in a sense, a radical proposal of Sherrington at the time. He's saying, really, because we're thinking now about evolutionary fitness, it's actually quite appropriate to think about the, quote, purpose, not as if there's some designer up there, you know, choosing how things will work because we need to get something accomplished, but because if you evolve a way of solving an important problem for the animal, you're more likely to survive. So it's appropriate to think about purposes, and in fact, reflex action cannot really be intelligible to the physiologist until she knows its aim. Okay, so this is a worthy pursuit here. So the question is, how do we go about thinking about this in the context of vision? Sherrington didn't really help us out here. But the people who did, I think, were the neuroethologists, because what they were studying were very characteristic instinctive reactions of animals to visual input that could be studied, reproduced, and ultimately they felt had some sort of biological underpinning, some genetic and ultimately neurobiological underpinning. So here's one good example of this. Some of you already, I can see smiles there, so some of you have seen this video before. So I want you to look at what the frog is doing here. It's orienting. It's seeing this black object moving against an otherwise stable ground, orienting and leaping. So this is food for the animal, right? This is how it survives. This is a useful visual reflex for keeping it alive. But be careful how you use it. So this kind of reflex response was well known to two important physiologists who were studying ganglion cells back in the 1950s, Jerry Letfin at the bottom left here and Horace Barlow at the top right. They were recording from frog optic nerve fibers. So they're recording the spike outputs of individual ganglion cells, and they found a variety of cells. So this is the early reflection of functional diversity of retinal ganglion cells. And one particular kind of cell that they found had a characteristic receptive field. It's a little different from the one that I showed you. The receptive field center was an on-off center, so either increments or decrements in light would cause the cell to fire. But around that was a region that would suppress the activity from the center if simultaneously illuminated. So a small spot flashed on or off in the center or moving within the center was good. But if you had global motion, for example, if the whole animal moved and the visual world swept across the retina, this is not going to activate this cell very well. So you could think about this as a object motion detector or a local detector of a stimulus in the world that was distinguishable from motion produced by the animal's own behavior. So I won't go through the quote here, but both Letfin and Barlow felt that this might be viewed as a fly detector or something like that, a bug detector. This is the terms that are thrown around. And this may be pushing things a bit far, but in the context of something that's hardwired like this, I think it makes a certain amount of sense. So Letfin actually was interested also in how this worked at the level of the brain. And here you see him lecturing in the 1950s. And what he's pointing at here is an image of the frog's brainstem, which I'm clearing up for you here a little bit. And what he's pointing to is this green structure in the dorsal surface of the midbrain, which is the optic tectum, or the superior colliculus, which is the major target of retinal output in the frog. So these centers in the midbrain are receiving direct input from the retina, including from these funny bug detector cells. And of course, what we know now is that in the superior colliculus, which is a major target of direct retinal input, there is a systematic topographic mapping of the sort that Descartes had already envisioned. So single points on the retina project their axon to single points within the tectal map. And ultimately, then you have a very precisely organized retinotopic representation of events in the visual world. Well, where do we go from here? Well, it turns out that in the deeper layers of the tectum, you have neurons that are connected to centers of the brainstem that are responsible for constricting the eye muscles, the extraocular muscles. So this is a way of redirecting gaze. And depending on the organism, it may also involve head movements or whole body orientation. So there's a motor map down here in the deeper layers. And this is systematically organized so that neurons in one particular place in the map will fire before reorientation to a particular region of visual space. Now, this superficial layer visual map and the deep layer premotor map are in alignment so that a given stimulus will activate a patch of the superficial visual map, which sits above just those cells which need to be active for the cell, for the organism to make the appropriate orienting movement to now align its gaze or its body towards that external object. So you can think about this as a fixation or a gaze-shifting machine. And this works for many sensory modalities beyond vision as well. But in the context of the present conversation, this is essentially the way in which the so-called bug detectors projecting through the tectum are leading to that orienting movement that you saw the frog make a minute ago. Now, this is the one point at which I can make a link to John von Neumann, because Letfin was part of a very interesting crew of characters working at MIT at this time, including Walter Pitts and Warren McCulloch, who were, as all of you know, the forerunners for all of computational neuroscience and neural network modeling. And they were building the simplest, earliest forms of neural net at this time and trying to understand what they might do for visual processing. Now, von Neumann was very excited about all of this, but he was also somewhat skeptical about how far you could really take this. And the quote that I extracted from a letter that he wrote to Norbert Wiener, who was another character, obviously, who was in the mix here, was responding to McCulloch and Pitts's work, was that the situation is rather worse than better than before, meaning before the first neural network models. McCulloch and Pitts have demonstrated that anything and everything can be done by an appropriate neural mechanism, and by neural mechanism here what they mean is a modeled, computationally modeled neural mechanism. So their models could compute, that was clear. But nothing we may know or learn about the functioning of the organism can give, without microscopic cytological work, any clues regarding the further details of the neural mechanism. In other words, you can build a neural net that will do something, but if you really want to understand how the brain does it, you got to get your hands dirty. So I take a certain amount of satisfaction in that, since that's how I spent my whole career. Okay, so that was a digression. Let me remind you that I've talked about one visual reflex so far that links a particular class of ganglion cells through a certain pathway to an appropriate motor output. Now, for that reflex to work, which obviously requires information about local object motion as distinct from global motion, image quality is essential, right? So if you've got a blurry image, the system is not going to work because any motion is going to cause some amount of movement over a wide retinal area, and the cells that I showed you before will simply shut down. So image quality is important, and there's a second visual reflex that helps to ensure image quality, and that's the pupillary light reflex. So what you know, of course, is that as light levels increase, your pupil stops down, and the advantage of this, of course, is that it increases your depth of field. This is like the f-stop on a camera. As you increase the f-stop, things that are not perfectly in focus are reasonably in good focus, whereas if you've got an open pupil, like in the top images, things that are in focus essentially only at the focal plane. In front and behind that, things get very fuzzy. So the operation of the bug detector system requires image quality, and that requires a pupillary light reflex. The reason you need a reflex like this is that, of course, as you stop the pupil down, you're letting less light into your eye. So if you're in dim light conditions, you stop your pupil down, now you won't have the sensitivity you need to see things, right? But in bright light conditions, you can afford to stop the pupil down very efficiently. So there's a sweet spot for any given lighting condition where you have optimal image quality and sensitivity. So the reflex does this for you. But how does it work? What does it need to know in order to generate the appropriate pupillary constriction? Obviously, it needs to know how many photons are entering the eye per unit time. This is what we call irradiance. It's light energy striking the retina. Okay, so the more light energy there is, the more the pupil stops down. And this is a reflex that's mediated through the brainstem in response to input from the retina. Now, it turns out that the pupillary light reflex is really only one of quite a number of reflexes or behavioral responses, hardwired physiological responses, that are related to the absolute light intensity as distinct from contrast boundaries within the visual field, movement pattern, or whatever. Okay? So among these are the entrainment of your biological clock to the day-night cycle. I'll have more to say about that in a minute. The amount of melatonin circulating in your bloodstream, sleep-wake cycles, alertness, and of course, very bright light can be aversive, so we blink. People who have migraines are photophobic, so it's painful for people and so on. All of these relate not to the structure of the image, not to any object information or pattern or color, but rather to simple light intensity. Now, back around the 1990s, it became clear from the work of Russell Foster and his colleagues that in animals with severe degeneration of the outer retina, where the rod and cone photoreceptors live, many of these photic responses to bright light persisted. Now, if the eyes were removed, that fell away. So it clearly suggested that somewhere in the visual system, there might be another photoreceptor. And maybe the most spectacular demonstration of this came from work on the circadian system. So let me just sort of walk you through this for a minute. What you're looking at is a plot of activity over a 24-hour period. Actually, it's probably more like a 48-hour period. So an animal is in a dark light cycle, dawn and dusk in the laboratory environment, essentially by turning on and off the lights. And what you can see in this animal, which is actually a mouse, so it's nocturnal, is that most of the activity, marked by these little vertical tick marks, occurs during the dark period. That's when the animal is active, searching for food. In the light period, it's inactive, largely sleeping. Okay? So there's a very close constraint of the activity to the day-night cycle. Now, if you plunge the animal into constant darkness at this point, so you're going through days here now in the dark, what you see is that there's still a consolidation of the activity pattern, and it still has about a 24-hour cycle. But it's not quite exactly 24 hours. It's drifting a little bit. It's like 23.5, so it drifts a little bit every day. So this is the free-running circadian rhythm. There's still a rhythm that's generated by a part of your hypothalamus known as the suprachiasmatic nucleus, but the exposure of the animal to light cleans up the clock. It takes it from 23.5 hours a day to exactly 24 hours. So now the animal is entrained, we say. Now, if you take the animal while it's in constant darkness and you pulse it with light at an appropriate circadian phase, what you'll see is this jog in the onset of the activity. So that's another indication of the effect of light on the circadian clock. How does this effect come to be? Well, it turns out that there's a direct projection from the retina to the essentially clock chip in your brain, the little nucleus in the hypothalamus that actually serves as the circadian pacemaker. We know it's the pacemaker because if you take this structure out of the brain and put it in a dish, it's still cycling at 24 hours. It doesn't need any other input. It's actually a molecular biological feedback circuit that's causing this thing to oscillate with a period of about 24 hours. So it's this retinal input, the retinohypothalamic tract to the hypothalamus, that is responsible for this entrainment, for this perfect alignment of the activity rhythm with the external world. Now, what I've told you is that in animals with severe degeneration of the outer retina, this process still works. If you take the eyes out, it doesn't. So this suggests that although we've thought that the only photoreceptors in the eye were the rods and cones in the outer retina, there has to be something else. And a key breakthrough came in the year 2000 with the discovery of a novel photopigment gene. It was named melanopsin because it was first found in the dermal melanophores of frogs, but it was later shown to be present also in the retinas of mammals. And in mammals, it's found only in a very small population of cells in the inner retina, in the layer of the ganglion cells, suggesting that this might have been the missing photoreceptors. This is what has inspired us in 2002 to go after the cells that were feeding the suprachiasmatic nucleus, this clock chip region of the hypothalamus. And we did that by injecting retrograde tracer into this region of the brain in rodents and tracing back to the cells of origin of the retinal input to this clock region, and then targeting those cells for recording. And just to make a long story very short, the answer is that in fact those cells are directly photosensitive. You can see that in the top trace. You can see the spiking evoked under conditions where all retinal synapses have been blocked. Okay, so this little step here is the increment of light, and you see this causes a depolarization in the cell, and it's spiking away. In the bottom trace, you're looking actually at recordings made from a cell that has been isolated from the rest of the retina. So we take enzymes and dissociate the living retina and record just from the red-labeled ganglion cell that innervated the clock, and sure enough, that cell will give you a nice inward current, an excitatory current in response to light. So this is a third retinal photoreceptor, but oddly, it's a ganglion cell that also talks directly to the brain. Now, these cells have some interesting features that suit them very well to their role in detection of light intensity and regulation of the circadian clock. The first is that they're unusually sustained in their response to a step of light. So most ganglion cells, shown in red, will adapt rather quickly. And this is because most ganglion cells are interested in change, either over space or time, in light level. By contrast, these intrinsically photosensitive retinal ganglion cells, that's what the IPRGC acronym refers to there, do adapt a bit, but they really hold their response for very long periods of time. In fact, a former postdoc in the lab has shown that they'll hang up there for at least 10 hours. When it comes to encoding the number of photons raining down on the retina per unit time, most ganglion cells are extremely poor. So on the x-axis here, you have increasing intensity, and the schematized ganglion cell is really not showing much change in its firing rate over time. These funny photoreceptive ganglion cells, by contrast, monotonically increase their firing rate as a function of light intensity. They are encoding the very feature that we suggest is necessary for these irradiance-driven responses. And then finally, whereas most ganglion cells have this center-surround organization that I described earlier, which is reflected by a sort of inverted U function. As you increase spot size, you get more and more response out of the cell until it fills the receptive field center. And then as you increase the spot beyond that to encroach on this surround region, you now begin to suppress the cell's response. So there's an optimal size for the spot to activate the cell. In the case of these ganglion cell photoreceptors, you essentially see no evidence at all of surround antagonism. And this is part of a general strategy these cells seem to operate under, which is to essentially summate photons over space and time. They're not differencing. They're not interested in contrast and structure in the scene, but rather in just the number of photons. How do they get to be so good at this? What makes them different? Well, one of the things that clearly is different is that their whole phototransduction biochemistry follows a logic very different from that in the rods and cones, and instead looks much more like what you see in the photoreceptors of insects and mollusks and so forth, the invertebrate photoreceptors. And I won't have time to go through all of the ways in which these are similar, but the ganglion cell photoreceptors are shown in the right column, and they line up with the so-called rhabdomeric or invertebrate-like photoreceptors in many, many respects as shown here. and not at all with the so-called ciliary photoreceptors, the rods and the cones. So in particular, one thing that may be relevant here is this issue of pigment biostability. So the rod and cone pigments are composed of a protein that binds a vitamin A derivative, and when that photopigment absorbs light, it causes a configurational change that leads to the light response in the photoreceptor, but it also bleaches the pigment. So you get a dissociation of the chromophore, the vitamin A derivative, from the protein. That doesn't happen in rhabdomeric or insect-like photoreceptors, and apparently it doesn't happen in the melanopsin, that is the photopigment of iPRGCs either. So this suggests that under constant bright illumination, the bistable pigment is constantly flipping back and forth between an excitable state and an excited state, and it doesn't require ancillary tissues or slow enzymatic processes to reconstruct the photopigment. It's happening in real time all the time because of absorption of light. That's clearly part of a story that happens at the level of synaptic inputs to these cells. So what you've seen already is that the canonical visual circuit is leading from classical photoreceptors through a synapse onto the bipolar cell, and then from the bipolar cell to the ganglion cell. This is occurring in the so-called inner plexiform layer, the major synaptic layer where bipolar cells convey their signals to ganglion cells. Now these funny ganglion cell photoreceptors, like other ganglion cells, have their dendrites in the synaptic layer, so they are likely to be getting synaptic inputs, and in fact we can show that that's true by eliminating the direct photoresponse through the melanopsin process, or by simply dropping the light intensity. So this is an intensity series, brightest light at the top, dimmest light at the bottom. At dim light intensities, the melanopsin system is not actually engaged at all. The threshold is quite high for that system. So at low light intensities, you're essentially looking at synaptic drive to the same ganglion cell through the rod and cone to bipolar circuitry. So even though the cell can respond to light on its own, it actually does also blend signals from the classical photoreceptors. Now, there's something peculiar about the contacts between the bipolar cells and this new funny ganglion cell. And that has to do with the sign of the response of the ganglion cell to light. So we know that there are basically two kinds of ganglion cells, on cells and off cells. On cells respond to increments in light, offs to decrements in light. And this is a reflection of the bipolar cells that drive them. This is occurring in two different layers of the synaptic zone. The off cells send their dendrites to the top and get input from bipolar cells that are excited by dimming or inhibited by light. And the on-center cells are stratifying here, and they're getting input from a class of bipolar cells that has the opposite sign of response. So essentially, you have a segregation of these on and off channels. Now, what you can see here is this ganglion cell photoreceptor is stratifying in what should be the off sublayer. It should be getting inputs from the off bipolar cells. And yet you can see that it's an on response. It's excited by light. So it's completely breaking the rules that we all thought applied to the on and off channels of the retina. So it occurred to us that one possibility was that these cells are in fact receiving inputs from on bipolar cells, but at synapses in a place that they really shouldn't be making any, in the upper part of the plexiform layer. And in fact, that proves to be true. I won't have time to show you much of the data, but this clearly is part of what's happening here. So I'm just going to show you now an image taken from some electron microscopic work that we've been doing on such synapses. And in this image, what you're looking at is a green bipolar axon passing by the dendrite of one of these melanopsin cells. And right here, you see this very strong synaptic specialization. Each of these arrows is pointing to what's called a synaptic ribbon, which is a structure that is really highly specialized for continuous, fast release of synaptic vesicles onto the postsynaptic target. And we suspect that this very unusual synaptic arrangement is a part of what makes the melanopsin ganglion cells so good at irradiance coding. One other peculiar aspect of this arrangement is shown here. So if we blow up the schematized synapse between the conventional bipolar cell and its ganglion cell target, what you can see is an arrangement in which you have two postsynaptic partners. Here's the synaptic ribbon. These are synaptic vesicles. And here's the ganglion cell that we're interested in. But in addition, there's a second partner. This is a process of an amacrine cell one of these inhibitory interneurons, and it's known that at these dyad synapses, the inhibitory interneuron driven by the bipolar cell is feeding both back onto the bipolar cell and forward onto the ganglion cell. And what this means then is that you have the potential for temporal filtering. Essentially, if you had a sustained drive, shown in green here, from the bipolar cell to the ganglion cell, but then you also use that same drive to drive the amacrine cell, and that amacrine cell is now inhibiting, either pre or postsynaptically, this circuit through to the ganglion cell, then what will happen if you sum this delayed inhibitory sustained input with the direct excitatory input is some kind of transient response, which then sort of renormalizes. So this is a temporal filter, a high-pass temporal filter, you could say, a change-sensitive circuit. By contrast, what we have found, and others have reported as well, is that these peculiar synapses in the upper part of the synaptic layer, these funny synapses I showed you in the EM, the electron microscopy a minute ago, are what are called monad synapses. There's only one postsynaptic target, in this case the melanopsin ganglion cell, and the absence of this inhibitory circuitry may mean that the sustained drive can pass all the way through the circuit without the temporal filtering that is typical of other ganglion cells. Okay, third reflex, and this may be the most challenging. This is a little hard to watch, but just see if you can see any butterflies flying through the air here. Anyone see anything? Okay, let's do this again with image stabilization, right? So the handheld camera is now, see the butterfly there? It's the same images you saw before, but the handheld camera was causing so much jostling. There's so much motion in your visual field that it actually made it difficult for you to detect the motion of this white butterfly going by. So image stabilization turns out to be an extremely important feature of visual systems, visual motor systems. And you can see this in all kinds of species, and this is one of the things you can do in your backyard if you're one of those urban chicken raisers. But basically, in this case, the animal is detecting the motion and using its head to stabilize the visual scene. For us, this is usually handled instead in response to self-motion that involves head rotation. So as I move my head to the right, my eyes are counter-rotating to the left by the same angular amount, and this stabilizes the image on my retina so that I can detect motion out in the visual world. Most of this is being handled by the vestibular apparatus. So you probably know the vestibular apparatus detects head rotation through the semicircular canals. There are 3 fluid-filled tubes, and these detect essentially the relative motion of the bony canal and the fluid inside. So if the head snaps briskly to one side, the fluid stays behind while the bone moves. The semicircular canals transduce this and use this to drive a visual reflex that stabilizes the eyes. So here's the basic sort of structure of one of these canals. The best axis for rotation, of course, is the axis that passes through the plane in which the canal sits. There are 3 canals, each with its own axis. So if we take the canals away and just look at the axes, these are the cardinal axes of the vestibular apparatus. 3 mutually orthogonal axes of best rotation for activating these 3 systems. So any arbitrary rotation of the head, of course, can be unpacked by the brain as the relative activation of these 3 channels. Now, this system works great as long as the head is moving quickly. So on the bottom axis, you have head velocity. The faster the head moves, the better the system works. So the gain of the vestibulo-ocular reflexes, which is what this is called, the amount of eye counter rotation for a given head rotation, is a function of head velocity. So at high head velocities, pretty good. As the head rotates more slowly, the fluid does not move with the canal. I'm sorry, the fluid moves with the canal, so you don't get any shearing force. You get no relative motion there, and the system breaks down. What that means then is you're going to end up with residual slippage of the visual scene across the retina. That's bad for perception, okay? So fortunately, the retina itself detects this motion. Essentially, the error, the shortfall of the vestibular apparatus results in a visual motion signal. It's a global visual motion signal, retinal slip, that can be used by the nervous system to correct for this. So if you fix an animal's head so it can't move its head at all, the vestibular apparatus is not in the picture, and then you sweep bars across its visual field as shown here, you can see that the eye tracks slowly and then resets. This is a nystagmus. It's exactly the kind of thing that you get with vestibular drive. It's the same motor apparatus, but now what's driving this is a visual input, which is what's left over from the slop of the inadequacies of the vestibular apparatus. Now, it turns out that the tuning of these visually sensitive elements in the eye is complementary to that of the vestibular apparatus. It works best for slow velocities of visual motion. And because these two are converging onto a common motor output frame, the sum of these two together give you essentially perfect image stabilization across the full range of head rotation velocities. Okay, so how does this thing work out in terms of synaptic networks in the brain? Well, we have the vestibular apparatus driving the vestibulo-ocular reflex, the VOR, through its connections to the vestibulocerebellum and brainstem, and from there we get access to the oculomotor plant and the extraocular muscles for eye counter-rotation and image stabilization. What we know is that there is a visual analog, optokinetic nystagmus, that I've just shown you, and that the key site for the source of this visual information to the vestibulocerebellum is a part of the brainstem called the accessory optic system. The details don't matter. It's a brainstem center that receives direct retinal input, and one would imagine it's carrying signals about retinal slit, this global motion of the visual scene across the retina. Now, in the 1980s, a guy named Jerry Simpson working at NYU made an absolutely brilliant insight, which was that the organization of visual motion signals in the accessory optic system was what he called a visual system in vestibular coordinates. What he meant by that is that the visual motion representation in the system was designed to detect rotation of the head around the same axes used by the three semicircular canals. Okay? What does that look like in terms of a visual receptive field? So here's a rabbit sitting in an imaginary visual world, and what Simpson found in the accessory optic system were cells that responded to visual motion in many regions of the visual field. This could be a whole hemifield where it was sensitive, or it could be even panoramic, both eyes. But the direction of motion preferred varied according to where it was tested. So in this case, it preferred up in the front part of the visual field and down in the back part of the visual field. Okay? So you can see quickly that the best stimulus for activating this cell would be rotation of the visual world around an axis. Now, the amazing thing that Simpson showed was that the axes that emerge from an analysis of complex visual motion receptive fields like this were identical to the ones used by the vestibular apparatus. This is a vestibulocentric rotational motion visual system here. It's essentially a visual system designed to speak vestibularese. Okay? So Simpson found cells not just like the ones I showed you, but cells that matched or preferred the optic flow arrangements for rotation around each of the three semicircular canals. Okay? So we have a visual analog for each of the three semicircular canals here. Now, how is this system built? Well, what's very clear is that the inputs to the system come from a subset of retinal ganglion cells that are selective for visual motion and are responding only at light increments. These are the so-called on-direction selective ganglion cells. And classic studies suggested that they came in three flavors: cells that liked upward motion, downward and slightly backward motion, or forward motion. Okay? Three channels, sounds sort of like the three vestibular channels, but we actually have a bit of an issue here if you think about how this thing might be constructed. If you project those directions of motion preferred by individual cells out onto the animal's visual world, it would look something like this. Okay? So either liking upward, downward and backward, or forward motion. Three types. Each one has a relatively small receptive field. It's looking at a small part of the visual field, and arrayed across the whole visual world, it would look like this. We're trying to build a cell that likes this. Okay? It likes rotatory motion around some axis, pointing out at you this way, okay? One of the vestibular axes. Well, let's overlay these things. how could this work? Well, perhaps a neuron in the brain that has one of these rotationally sensitive receptive fields is selectively sampling from different populations of direction-selective cells in different regions of the retina, weighting the relative inputs from these different channels so as to reconstitute this rotationally sensitive receptive field. That could work. But we had a radical alternative hypothesis, which is that the earlier studies had gotten it wrong because they'd only really recorded it one place in the brain. That instead, it might be that one type of direction-selective ganglion cell in the retina actually adjusts its directional preference as a function of where it sits in the visual field, that is, where it sits on the retina, so as to always match the optic flow that would be generated by rotation of the head around one of the semicircular canals. Why would that be useful? Well, if you imagine a neuron in the accessory optic system that gets convergent input from all of those cells across the entire retina, what you end up with is this kind of receptive field. Okay? So the key thing here is the suggestion that the earlier argument that the directional preferences of individual ganglion cells is fixed. That's the same directional preference regardless of where we record a cell of that type on the retina, that instead what you have is a population of neurons that in the ensemble have a directional preference that matches the rotatory field. But any individual cell might have a very different directional preference for one of its compatriots that sits in a different region of the retina. Essentially, the computation by this analysis would be done in the retina itself. So Shai Sabah, who's here, postdoc in the lab, has been testing this crazy hypothesis using calcium imaging in mouse retina. And what you're looking at here is a plot on a flattened mouse retina of all of the cells he's recorded so far. It's a heroic task. It's like 500 cells or something like that. And remember that the classic model suggests that there are three directions of preference. Now, if you look at those arrows, I challenge you to find those three directions in there. It's a mess, right? It doesn't look anything like the classic model. In fact, if we reproduce what the classic folks have done and sort of align all of these different directional preferences at a single point, what you see is every possible direction is represented, and if anything, what you see is maybe a weak suggestion of four preferred directions. So it's quite different. Now, this is enough to, I think, throw the old model out, but is it enough to tell us that this flow matching idea is right? Maybe, maybe not. There's a lot of evidence on this. Shai could tell you about it if you want to hear more detail. I'm going to just show you one line of evidence. And the idea is this. If you want to figure out whether this complex representation, it has embedded within it multiple flow matching sort of rotatory cell populations, if the underlying logic here is rotational, then what you ought to be able to do is start with this distribution of cells, pick an arbitrary axis of rotation, and reconstruct the optic flow that would occur if the head rotated around that axis. So that's what's being shown here in the blue lines. We've got flow fields on the flattened retina as they would occur if the head rotated around an arbitrary axis. This center of rotation corresponds to the projection of that axis out into the visual world. Okay? Now the question is, how well do the directional preferences of individual cells we've recorded in the retina align with those flow fields, right? And we can assess that by looking at the distribution of difference angles between the observed preferences of the individual cells and the prediction for a particular flow field. So that's shown here, and you can make a histogram of these difference angles, and if you've got a pretty good alignment, you might expect a piling up of observations here, and we see that in this particular case, and maybe see fewer at other orientations, right? So for any given axis, where we're testing the fit of the actual observed directional preferences to the flow fields, we can assign an index of goodness of fit and essentially have one point in a heat map, a mapping of the goodness of fit across the retina. Now we have to redo this for another axis and yet another axis, and do this for axes separated by 10 degrees in azimuth and elevation, and then take a look at the heat map. So what I'm going to show you in the next slide is a heat map generated that way and wrapped back onto a sphere. And if there's a rotational structure to this directional preference distribution, then you should see clear differences, hot spots and cold spots. And this is what you see. So clearly there's a lot of structure here. And it's very hard to get your mind around what you're looking at here. I understand that. But the big gaps between hot and cold here and the numbers of hot spots are precisely what you would expect if, in fact, our hypothesis was correct. That is, if the directional preferences of these direction-selective ganglion cells feeding this image stabilization network are actually already in vestibulocentric coordinates. They're matching the flow fields that are generated by rotation of the head around a particular semicircular canal. So you just have to take it on faith. If you can't work through the logic here, as I imagine it would be tough to do. Okay, so just to give you one last look at this, what I'm showing you here are three versions of that heat map, overlaid on top of which you see the optic flows that would be associated with rotations around each of the three vestibular semicircular canals. And what you should be able to see is that where the pole for each of those rotational systems sits, that is, where there's a center of rotation, you tend to find a hot spot nearby. So for this rotational system, a hot spot here. For this rotational system, a hot spot back here, a little bit hard to see at this orientation. And for this last one, it's a little bit weaker, but there's some heat in here as well that presumably corresponds to that third canal. So what I've tried to do by way of sort of pointing a way of thinking about how to break down the complexity of the ganglion cell population and the functional specializations of those cells and their projections to the brain. We've gone through three different visual reflexes. On the left, gaze shifts and probably even attention shifts to objects in the world based on real-world object motion. The system I just talked about, the image stabilization system, and then on the far right, the pupillary light reflex is one example of an irradiance coding system. In the table, I've tried to make the point that different ganglion cells are feeding these different systems. On direction-selective ganglion cells for image stabilization, melanopsin cells for the pupillary light reflex, and these bug detectors or local edge detectors, as they're sometimes called, for object orientation to objects. Now, the spatial precision of these systems varies dramatically across the board. So for the collicular gaze-shifting system, spatial precision is essential. For the pupillary light reflex, this melanopsin ganglion cell system, not at all, and fairly high also for the image stabilization system. Retinotopic organization also varies radically. This is kind of hand in glove with spatial precision. So again, very highly topographically organized system for gaze shifts to objects. In the case of the pupillary light reflex, massive spatial and temporal conversion. So that's kind of irrelevant in that case. And then for the image stabilization system, a funny kind of hybrid, because of course the individual neurons have small receptive fields and prefer direction of motion in one particular location in the retina. But ultimately what you're getting to is a global representation. You get these panoramic visual receptive fields that are driven by inputs from neurons of this type across the entire retina. That's how you're building the global representation. So is that precise or imprecise? It's a little bit hard to say. There are differences in these systems in terms of temporal integration, whether they're driven by the on or off pathway. I haven't had a chance to talk about it, but their dendrites sit at different levels in the interplexiform layer, so they're presumably drawing inputs from different classes of bipolar and amacrine cells. And finally, their relationship to the oculomotor system is kind of intriguing, because the gaze shifting system is tapping into a rapid eye movement network in the oculomotor system, very rapid shifts of gaze. The image stabilization network is sort of classic slow eye movement system like you would see during smooth pursuit. And even the pupillary light reflex system is linked to the oculomotor system, but now it's the intrinsic musculature of the eye, in this case, the iris muscle. Okay, so we're supposed to think ahead 250 years. Leon would like us to know sort of where we're going to be 250 years from now. I shudder to even make a guess at this, but I think we can point in a couple of specific directions. The first is with the advent of things like serial block face electron microscopy, an image of which you saw earlier. I think we are very much on the verge of being able to fully reconstruct synaptic circuits in at least restricted regions of the brain. That's today. So give us 250 years, I don't think there's any question at all that we can completely reconstruct all of the synaptic connections of all of the cells of known type everywhere in the nervous systems of experimental animals, and maybe at least in postmortem human material. Whether we could get that level of specificity in the living brain, I have no clue. That's a great question. So storage is going to be a problem going forward. So this is just an example of what's already been accomplished in mouse retina. For a small volume, every cell type, every example of every cell type and its synaptic connections have been worked out. We have fantastic markers for specific cell types. We know to some extent which genes are expressed in which cells. We're just seeing the dawn of that now. We'll have that in extraordinary detail, I think, in a matter of decades for virtually whatever cell class you want in the mammalian nervous system. We have the ability to use genetic methods to kill cells, to excite cells, to silence cells, to introduce specific proteins into specific cells. This is all now. So 250 years down the line, the amount of control that we're going to have over the brains of at least experimental animals is going to be phenomenal. And of course, we're already getting into the realm now of therapeutic approaches that exploit this kind of experimental power. So for example, we now have animal models that have severe outer retinal degeneration. We can introduce virally proteins into cells that would not normally be photoreceptive, turn them into photoreceptors, and actually recover visual function in animals that were functionally blind before. We can cure single gene defects in human blind patients now. So the future is really promising. I think in 250 years, it would be shocking to me if it weren't the case that we could completely cure, or at least ameliorate, any kind of functional blindness through methods like this. I think, just as these days a cataract is no longer a cause for blindness because you can take the cloudy lens out, I think the same thing will be true for degenerative retinal diseases as well. I think what's even more mind-bending, but maybe not beyond the realm of the possible, is that with the advances in methods for controlling the activity of neurons in living brains, that it may not be simply a therapeutic sort of approach that is being used for these kinds of things, but rather that it may even be elective, that it may be that sort of the advent of wearable computing is going to quickly get into the realm of implantables, and that the most remarkable heads-up display that you could ever design is going to end up being your own visual cortex. So that would be my guess for 250 years down the line, Leon, would be that in the end, you want a virtual reality system, it's going to be the manipulation of your own cortex that's going to be creating this experience for you. So with that, I'll give it up and take some questions, if you will.